Carney, CEO of CRISPR, with us. With that, Sam, maybe starting a picture on the strategy here. So you have, cancer, ex vivo non-cancer, regenerative medicine, in vivo verticals. How do you think about capital allocation across these different verticals and where you, at this point, as you kind of de-risk a vertical, how you kind of go deeper into it?
Yeah, thank you, Salveen, for having us. It may be hard to believe for many of you, but we're in the 10th year of our journey as a company. We follow the classic formula, sort of, which is say, let's pick a very good indication where the technology can be de-risked, where there's a great unmet need, and there's an advantage or competitive advantage with the technology we're deploying. And we did that with sickle cell and thalassemia. And the next thing we did right after that is deploy those learnings to diversify into other platforms. So for those who are new to the story, we have 4 verticals right now. One is hemoglobinopathies, where we have an autologous cell therapy. We have immuno-oncology, where we not only have our first gen, but our next gen programs. These are allogeneic CAR Ts going after both heme malignancies and solid tumors. We've then expanded to regenerative medicine, where we use iPS cells, edited iPS cells, and apply that towards diabetes, particularly type 1 diabetes. And our 4th pillar that we've put together now in the last 2 years is our in vivo pillar, where we hope to get into the clinic this year. And with our in vivo pillar, we're going after cardiovascular diseases to start with, but there are many other indications coming beyond that. And what that's done is given us a broad-based portfolio where we have, obviously, the technology linkage, but they're independent bets from a biological standpoint or validation standpoint. And that gives us great confidence that we can not only expand in the area where we feel very good about right now with exa-cel and hemoglobinopathies, but well beyond that, and gives us tremendous upside potential in the next year or two. So from a resource allocation standpoint, obviously more of our spend right now is towards our more mature programs like exa-cel. But what's more important for me is the amount of time spent. And I think, if you ask me where I spend a lot of my time, I'm spending a lot of my time with in vivo vertical, getting that up and running. The 5th pillar, if you will, is CRISPR X, which is our next gen editing technologies. And we're spending a lot of time there actually putting that forward. And that's showing all sorts of interesting data, whether it's correction, long-range insertion into genomes, et cetera. So all in all, we're very pleased with the breadth of our portfolio, and we feel like we have enough resources to do this in an efficient way and push all of them forward.
So maybe jumping into this in vivo vertical where you've chosen to go into cardiovascular disease, help us understand why that was the first approach in the context of, the risk-reward profile, right, where this could be a population where people wait a little bit longer and want to monitor, off-target edits or any aspect that could play out there. And then it's also been a very difficult payer environment in that market. So what made you think this is the way to kind of start and go with this vertical?
Yeah, so I think, it was a counterintuitive bet at the time. If you think about 2017, 2018, a lot of pharma companies were getting out of cardiometabolic. Now that's reversed. You're seeing every pharma company jump back with the success that we've seen with GLP-1s and some of the obesity and weight loss drugs. But it was an environment where, people thought that the investment profile didn't make sense. Large clinical trials were needed. While the PCSK9 story is a fantastic story from a science standpoint, there's a lot of resistance from payers initially. But what people need to realize about our portfolio, we have ANGPTL3 as a target, and I'll talk more about it, LPA as a target, that we're not trying to go directly into the broad population with tens of millions of patients. We're trying to go at, develop a strategy where we go into the rare populations. Let's say it's HoFH, familial hypercholesterolemia, whether homozygous or heterozygous. There are certain populations that have refractory to statins and all sorts of medicines. In fact, there are patients who are refractory to PCSK9 antibodies that would need something else, like ANGPTL3 or LPA. And so to find, the right mix here is biological validation. At this point, ANGPTL3 is very well validated. In fact, there was recent data from Arrowhead showing the cause effect of ANGPTL3 silencing or downregulation. The second part is there a development path that's somewhat lean and efficient that allows a biotech to take it all the way to label and then allows to expand beyond that. And the third is there a competitive advantage? And a one-time gene editing approach will have a huge competitive advantage. The compliance within this population, whether it's a 6-month injection or a 3-month injection, is very low because the disease is not visible. You're not seeing it like you're seeing in the obesity situation. It's you're not looking in the mirror every day and saying, "Oh, my arteries are clogged." So the compliance is very low. So one-time downregulation or deletion of the gene is very advantageous in the long run. The second thing is the payer environment. If you think about pharmacoeconomics, it would benefit the payer tremendously to do a one-shot gene editing approach because let's say it costs $250,000 for a one-time edit for one of these genes. The benefits are great and the value is there. But the second thing is you're not paying tens of thousands of dollars per year for an antibody or something or an siRNA. And so there's a pharmacoeconomic benefit as well for the system. So all in all, I think it's a complex calculation, but we feel very good about the approach that we have with ANGPTL3 and LPA. We'll follow that with PCSK9, which is a little more complex because the number of agents that you have with PCSK9 is many more and it's more competitive. But at least for ANGPTL3, we have a pretty clear path. We'll get more regulatory feedback as we go along. But right now, we're focused on getting into the clinic and dosing patients.
And then help us understand with regard to your toolkit, right? CRISPR-Cas9, clearly you've deployed it across all these different programs. But how are you adapting to the dynamic landscape just in terms of the future when you think sustainability? And what I mean is, we've seen base editing, we've seen gene writing, and how do you think about matching the tool to disease and then the need to evolve or bring in other technologies?
We're seeing everything play out very similar to what happened in the late 80s with antibodies. We had the original antibody technology and IP, and then people had improvements on it. Whether it was humanization of the antibody, it was fully human antibodies, smaller fragments, all that were improvements upon the original antibody technology. And it wasn't like one platform was better than everything else and was going to take over. It was just what was applicable in a certain setting. And over time, what we've seen is value ultimately accrues to the company that can actually develop drugs effectively and use whatever tools are available. And that's our approach. All these forms of editing that you're seeing, whether they're target prime editing or whether it's large insertion of RNA-based editing, using RNA-based editing, they're all improvements upon the fundamental concept, which is that you can take a Cas9 protein, put it together with the guide RNA and direct it to any particular region on the genome, like one locus in the genome. So you're targeting it there. So you have like a carrier of sorts on the genome, and then you can put any effector protein on top of it, whether you want to do a NIC, whether you want to do a double standard break, whether you want to do something else. So I think those are all improvements. And for us, we espouse all of them. In fact, that's one of the key aspects of establishing CRISPR X. We established as a company within a company that they're incentivized differently, they operate differently, and they're doing short corrections now with using RNA. They're developing new donor templates. They have new delivery technologies, and all that with the notion that we don't want to get disrupted. I think the classic, the 2 Ds of biotech, death by disruption and death by dilution. And we've seen that time and time again with companies that were high flyers in the space. And we want to avoid both. That's why we have a very strong balance sheet. But from a disruption standpoint, we want to be far ahead. All that said, I think you're getting to a point of marginal improvements in technology. I don't see, something coming along that's going to do to us what CRISPR did to zinc fingers or talons. I just don't think that kind of step change disruption is going to happen. You're going to see marginal improvements that make it more applicable across different disease areas, and that's great for the entire space.
And just remind us where you stand in terms of optimization around delivery. I mean, there's been a ton of work there for different tissue targets.
Yeah, what I would say is that's the space where we're seeing a lot of progress right now. Delivery using LNP to the liver, I think, is solved. I think there are many different LNPs you can use, and they're available out there, whether you develop it yourself or you can license it in. And I think we can get genetic material to the liver for RNA-based delivery. For double-stranded donor templates or for DNA delivery, there's still some work to do. For all the other organ systems, you're seeing a rapid improvement in the technology because there are ways now to sort of barcode or identify the LNPs and just try a different mix and empirically see what's going to which organ systems. So you're going to see LNPs get to other organ systems in the near future, including, let's say, hematopoietic stem cells or whatever else. But again, there's work to do. It's not, these things don't happen, within all of a sudden, they don't fall off a cliff and all of a sudden you have fruits of all the work you've done. It takes time to optimize, and you're going to see more and more opportunities emerge. There's also work being done with all the AAVs. I know the investor sentiment is soured on AAVs a little bit, especially given some of the regulatory challenges, but you're getting these second and third gen AAVs now that are very tropic to the brain, for instance, to muscles. And so for neuromuscular indications, I think it may still be in the AAV realm that we see the next breakthrough.
Well, let's jump into the portfolio here. And so with exa-cel, your gene-edited cell therapy for beta thalassemia and sickle cell, congrats on the progress. I know when we talked almost 10 years ago, I didn't think it would be this fast in the context of competition. But help us understand the economics and commercial agreement in place with Vertex and how you're thinking of approaching the launch in the United States in partnership with them from a payer standpoint, but then just a division of effort.
Yeah, I think the exa-cel story is quite remarkable. In fact, one of the senior R&D leaders who came out of Big Pharma is writing a book about the 3 or 4 drugs that were developed that are the most astounding to him, and one of them is exa-cel. And we went through the history of exa-cel, and the reason exa-cel moved this quickly, one is obviously the execution from both Vertex and us, but also it builds upon years of evidence. There were studies in the 80s done around population genetics that showed that elevation of fetal hemoglobin could render a patient or a person with sickle genetics normal, right? I think these are people walking around, and same with thalassemia. So you start with that basis, and then there's the basis of genetics that's happened over the last 15 years with various entities like Boston Children's and other labs that said, could you make a precise change in the genome to elevate fetal hemoglobin? And that theory is proven out as well, and that's shown to be able to be recapitulated in the lab, but also now in humans with exa-cel and other programs. But ultimately, I think for us, what allowed us to move exa-cel very quickly was starting with a clean slate on CMC. I think all the programs you've seen that have had delays with regulators has been around CMC issues. And I think that's mostly because it's been academic processes that are ported into companies that then have to be optimized. And you're putting Band-Aids on a lot of things versus starting off clean and saying this is a robust process from the get-go. And so we did that, and it allowed us to dose patients very quickly and get to this, I would say, historic point in time with exa-cel where we're in a position to be the first approved CRISPR-based therapy in the world. And for a platform to go from elucidation to fruition with an approved drug in the span of a decade has been unheard of in biotech or biomedical research before, but we are in that position. But work's not done yet. I think the work's only done when we get this therapy to patients across the world in a very scalable and smooth manner. And we're pleased that Vertex is leading the charge on commercialization. They have incredible experience commercializing rare diseases. They have experience with payers, not just in the U.S., but in Europe. They have experience establishing these centers and a very high-touch model around these qualified treatment centers. So we feel very bullish about how this could all play out. I think a lot of people are underestimating the level of demand there could be for a therapy like exa-cel. And it just underscored by our recent EHA data presentation, the amount of interest there was at EHA, the number of people who showed up for the Q&A later. It just tells you that there's tremendous excitement from the investigator community, but also just from a general physician community around an approach like exa-cel.
And remind us with the regulatory filing, have you provided enough follow-up and patient numbers in terms of what the FDA may require for a novel technology?
Yeah, I think, one of the benefits of having RMAT designation early on is that there was a lot of fluid exchange with the U.S. FDA, but also European agencies. And I think we've been trying to make sure that we have everything we need on CMC front, but also what we need from a package standpoint. But the data are irrefutable. If you look at the data, every patient has benefit. Every sickle patient, one patient who had a case of parvovirus or something like that, but again, that was only a temporary VOC. And every patient has had benefit. The data are very clear. These are patients coming in with several VOC episodes a year, and they're completely free of all the symptoms. Same with thalassemia. These are patients who are coming in with so much transfusion requirement, coming in not requiring that anymore. And people, underestimate and say, oh, it's transfusion, you can just get a transfusion. But for those who've gone through a surgery and have to get transfusion, it's pretty complex. You're getting someone else's blood in your system. There's, all sorts of side effects that happen, like inflammation, vasculitis, et cetera, et cetera. It's no joke that these patients live through that, and then they have iron overload. And if you can take these patients and get them free of all those symptoms and requirements, that's a pretty big deal for these patients. And hence we're excited about not just the prospects of the drug for us, but the prospects and what it means for the patients and this patient community.
And where do you stand from a manufacturing capability and capacity standpoint to address the demand?
Yeah, what we've done with Vertex is created enough capacity so we're not in a situation, at least in the early going, where we're supply constrained. And we've seen that happen with some of the CAR-Ts where they were supply constrained. And not only, build a lot of capacity, but have some buffer so that we can expand beyond where we are with how we utilize the existing capacity as well. So we feel good about that. Now, if we're completely surprised and the demand is beyond what we built, I think it'll be a good problem to have. But it's easy to add modularly more suites and more capacity along the way.
And then just talk about where you go from here in terms of going after the larger pool of patients. So, where does work stand with regard to a more benign preconditioning regimen or just even to an in vivo approach?
Yeah, I mean, both of those are priorities for us. We're working quite diligently on creating these conditioning agents. Our approach is based on c-Kit as a target with a toxin that's not as toxic as some of the other clinical trials we've seen out there. And with a different PK/PD profile where you get a quick hit. You want the conditioning agent to go in there, eliminate the stem cells from the bone marrow, but then not have any effect on the cells that are infused, which are the edited cells. So it's a different PK/PD profile, and we feel good about our approach because a lot of the other agents that were developed with AML in mind or oncology indications. So it's a different profile than built for purpose around sickle cell. So we'll continue to move that forward. I think we'll provide updates along the way. Now, Vertex have their own independent efforts, not just with c-Kit, but other targets as well, and a relatively large effort. And so between the two of us, if any of those work, I think that'll be have pretty big implications for the penetration of the market in sickle cell and thalassemia.
For these severe patients, and in particular the women of childbearing ability, are they pushing back on the ability on the willingness to take this drug, or are they gonna? Or how are you addressing that?
If the interest in our clinical trials is any indication, I think these patients are saying, I can't live with this disease anymore. I will take a chance, and I want to see a ray of hope around being normal and living like everybody else. And I think everything has risks. The transplant procedures have some risks, but compared to what they go through on a day-to-day basis and the life they live, in fact, I got to see we hired a couple of our employees had sickle cell disease and got to see what they live through every day. Every day they come into work could be the day you end up in the hospital, and it could be the day you end up in the hospital where you're treated like a addict and not get the opioids. And so they would carry these doctors' certificates and letters with them everywhere they went because should they end up in the hospital, they want to say, look, this is a legitimate need for painkillers and opioids. And I think these patients go through that, and when they see this chance that they could be normal, they're going to spring for it.
So let's transition over to oncology. It has been complicated when you look at what's been playing out in the allogeneic CAR-T space. CD19, it seems like, companies are there, but beyond that, there's optimization that's required, and you've talked about going to the second generation approach. So just to level set, help us understand where you're comfortable with the portfolios, which assets need more optimization, and when we could start to see more data over the next 12 months. Yeah.
If I zoom back out 20,000 foot level and say, if someone told me in 2017 that in 4 or 5 years we'll have data which shows that there's durable CRs in a heme malignancy with an allogeneic CAR-T first generation, actually that's a pretty good result. If you look at the history of some of the bispecifics, it's taken 15 plus years. If you remember the early BiTE data and the BiTE trials, a lot of work to get to this point. And allogeneic CAR-T is moving at twice the speed of bispecifics. And I think it's going to be better. The limit of improvement of an allogeneic CAR-T is a lot higher than the limit of improvement that you have with the bispecific or trispecific. And so we're pretty excited about what we've seen with the first-gen programs, 110 and 130. We're seeing a lot of interest in enrollment. In fact, at the EBMT conference recently, we had the presidential symposium with CTX 110. With CTX 130, we continue to see these stories where patients are wheelchair-ridden. They can't even stand up because there's cancer all over their skin, including their foot. and all of a sudden they're walking normally. And so the interest is there for the first gens already, and they, I think, can be their own valuable products in their own right. But then we're incredibly excited about the next gens. We came upon these two edits, regnase 1 and TGF-beta-R2. TGF-beta-R2 is more obvious, was been published before, but the synergy between regnase and TGF-beta-R2 was something that we saw in our empirical studies that struck us immediately. And we said, this is quite powerful. This is better than any other pairwise edit we can make in these CAR Ts, and they're 10, 20x better than the first-gen CAR Ts. And if that plays out to any extent in the clinic, all of a sudden we have not only best-in-class allo, but best-in-class products in the LBCL, let's say. And we also have the potential to make a dent in solid tumors. We were excited 2 years ago when we saw the first complete response in a solid tumor with an allogeneic CAR T. That was the first complete response with any CAR T, not just an allogeneic CAR T. And so I have to put all this in the context of scientific advancements, and I would say I'm pretty happy with how well allogeneic CAR Ts are going. And I know investor sentiment may be at an all-time low, but what's very encouraging to me is pharma is catching up steadily. All of a sudden, the level of interest from pharma has gone up in allo CAR T because everyone feels like, even the players that are not in cell therapy feel like they have to make a play in cell therapy eventually. They don't want to do it in autologous therapies because it's just not their bailiwick. And they want to be in a gross margin, profile of 90%, just like small molecules. And the only way to do it is allogeneic cell therapies. In fact, we think that we can get the COGS down to a level that's so much lower the second gens that we can make this an attractive product in China and India, for instance, and bring CAR Ts over there in a scalable fashion. So overall, I feel very encouraged by what I'm seeing on the allo CAR T front.
And when might we see data from the portfolio across the year?
Yeah, so we'll see continuing data for CTX 110 and 130 that are targeted towards CD19 and CD70. But right now, I think what we're very excited about seeing is the data with our next gens. Now, we may not disclose the data this year, but even with the early dose levels, what we're going to see is a difference in the PK/PD dynamics. In fact, we have a lot of pharma companies tell us, "Oh, don't wait for the data disclosure. Just call us when you see expansion data and PK/PD dynamics," because you're going to see very quickly, is this different from what you saw with the first gens? Are you seeing what you predicted in terms of expansion or the kinetics of the cells, but also what the phenotype of the cell is? Let's say you're at day 20 and you see a lot more memory phenotype CAR T cells. That's a very encouraging sign. So we're waiting to see all these data, and we're accruing and collecting the data, and we feel good about it.
Right. Regenerative medicine. So a lot of interesting development there with regard to type 1 diabetes and your partnership with Vertex on the ViaCyte side. How do you kind of compare and contrast that program versus the gene-edited SEMA program? And when could we get some early proof of concept from that portfolio?
Yeah, we're excited about expanding our partnership with Vertex into the regen med area as well. If you recall, the history of this is Vertex had a 50-50 partnership with ViaCyte. Vertex had a partnership with SEMA. They acquired SEMA. CRISPR had a partnership with ViaCyte. And last year, Vertex acquired ViaCyte. So they now have their portfolio of SEMA products. Effectively, the approach is the same, but the starting material is different. They have a cell line from SEMA where they have three approaches. One is naked cells without any edits where you require immunosuppression. They have the cells in a device where you may not need immunosuppression or may need it. And then they have the edited cells. And the holy grail here is to have these cells be infused into the patient without immunosuppression. And then all of a sudden you have a cure. You effectively have pancreas off the shelf that you've put into a patient that could render them free of diabetes, right? And on the edited cell side, the ViaCyte program that we've been working together with them on is far ahead. We've dosed a number of patients now to see what the data look like. We recognize that Vertex was going to have its own programs through the SEMA partnership. We actually licensed some of our IP around the edits to Vertex in a deal that we did earlier this year, with the notion that this is a high-risk venture, but the more shots on goal you have, the more you're going to learn. And eventually there's so much value at stake here with the notion of curing diabetes with artificial pancreas that we'll figure out a way to, divvy up the value or find the value ascribed to each party. But the more important thing is, how do we make this work for patients? And that's the single-minded goals that we have as CRISPR, but also I sense the same goal from the Vertex side.
Where are the risks with your program right now to kind of not achieving that today and needing more optimization?
Yeah, so we did our VCTX 210 trial early on with very low cell doses to alleviate some of the concerns that we perceive regulators may have, which is, are the cells coming out of the device? Is there anything else happening to the cells from a phenotypic standpoint? Are they fully differentiated? What are you seeing? So we did some of that. We have the data necessary to say we were confident moving forward to VCTX 211, and we're dosing patients here. So we feel good about the clone. We feel good about the edits we made in VCTX 211. The device engineering always needs more optimization, but ViaCyte had done a lot of that. This was their Gen 5 device in a way. So we hope that it just works with what we have right now, and would not need any more configuration or optimization.
What are you most excited about coming out of CRISPR X?
The ability to correct genes, but also to insert long genes into the genome in a specific fashion is going to be very powerful. What we've seen with the in vivo set of diseases they can approach address with in vivo approaches. There's about 20 or so indications you can do with gene knockdown right now. That's right in front of us. There may be more over time. But if you can correct genes or insert genes, that's like 100 or so indications you can go after. So the size of the prize in a way is much larger. The unmet need is also there in a lot of the indications. But I think we're pushing both forward in parallel. I think there's more biological validation happening. So if you take NASH as a disease, for instance, now we have markers and edits which are directly implicated in NASH, and you can just turn off those genes. You may have a potential cure for NASH, right? So that's all being validated through some of the clinical trials with siRNA, and we can quickly follow with gene editing if that proves to be positive. And then with gene correction, there are tens of diseases. There's the enzyme replacement type of indications like Fabry and Gaucher's. There's hemophilia. There's PKU. There's so many indications you go after if you can correct or replace genes. You can do that largely with liver delivery. So that's all exciting. So with CRISPR X, the mandate we have is let's be very smart consumers. We're going to look at all the technologies developed out there, whether they're RNA-based or DNA-based. We're going to look at all the donor templates out there. And what we want to see is correction levels that are meaningful for the indications we're going after, 20, 30%, with the appropriate delivery. We want to see insertions at a reasonable efficiency as well. And all these are data that we'll put out there at some point. I don't know if this year we'll do an innovation day or not, but at some point we want to say, here's our approach in vivo holistically with our advanced programs and everything else behind it, and here's the technology that's going to support 10 more programs in the future. So it's very exciting.
Any questions from the group?
Hi there. For your diabetes program, you mentioned the different starting materials that you use versus Vertex, and just wanted to see if you could walk through exactly what those differences in cell state is and the puts and takes of those two approaches.
Oh, it's just a different cell line. They had a starting cell line that Semi was using. That's, again, they're pluripotent stem cells, but it's just a different cell line. And you start with the different cell line, and then you make your edits, and you differentiate to whatever state you wanted to differentiate to. So that's the difference in substrate.
Well, with that, Sam, thank you so much. We appreciate the time today.
Yep, thank you for having me.